Hello, everyone, and thank you all for joining us during the Lytham Partners 2024 Fall Investor Conference. My name is Ben Shamsian, Vice President at Lytham Partners. During this webcast, we welcome Reviva Pharmaceuticals, ticker symbol RVPH on the NASDAQ. Joining us today from the company is Dr. Lax Bhat, Reviva's Chief Executive Officer. Before I turn it over to Lax, I want to remind everyone that management is available for one-on-one meetings throughout the conference. If you've not already signed up and would like to schedule a one-on-one, please send me an email at shamsian@lythampartners.com. That's SHAMSIAN@lythampartners.com, or visit our conference website. With that, Lax, the floor is yours.
Thanks, Pam, for giving us this opportunity to present here at Lytham Conference. I really appreciate it. So I would like to welcome everyone to our corporate presentation. Again, my name is Lax Bhat. I'm founder and CEO of Reviva Pharmaceuticals. We are a public company listed on NASDAQ, traded under ticker symbol RVPH. Before I present the corporate presentation, I would like you all to take a look at the forward-looking statement displayed here. Now, quickly move on to a very brief overview of what we do at Reviva and highlighting what we have recently achieved milestones and then upcoming milestones as well. So here is our development pipeline. So Reviva is a late-stage pharmaceutical company focused on developing next-generation therapies for diseases of neuro target. That's our focus and then also core strength as well. As you can see here, we have two molecules in development, both discovered in-house, and then company owns 100% patent rights for both the molecules, compositions of matter patents, as well as several methods of treatments as well. So the lead indication is schizophrenia. We have treated brilaroxazine in over 1,000 patients with multiple clinical trials that include large two randomized trials in schizophrenia and one long-term 1-year safety study that we are expecting to complete in the next couple of weeks. So based on the available data, we believe this can be readily developed for additional neuropsychiatric large indications, bipolar disorder, major depressive disorder, and ADHD. So based on the pharmacology profile of this drug, and then also the symptoms or additional comorbidities seen in this patient population, inflammatory conditions such as especially psoriasis, and then also these vulnerable patient population predisposed to pulmonary arterial hypertension, pulmonary fibrosis. Our drug has shown in the translational model robust efficacy for these inflammatory conditions, as well as in the clinical trial in schizophrenia. We have seen statistically significant reduction in pro-inflammatory cytokines. I will walk through some of the key data in this respect. So now to connect this inflammation and then psychiatric, here is a brief overview, and then a few points are highlighted here. The first thing is schizophrenia is not a single disease. Rather, it is a mix of multiple symptoms. In a major symptom domains are highlighted here: positive symptom, negative symptom, cognitive dysfunction, mood, and agitation. And then besides these 5 major conditions, up lately, what we are learning more about is neuroinflammation is real in every single schizophrenia patient. The degree of inflammation may vary from patient to patient, but neuroinflammation is found in every single patient. So our drug, the mechanism, as you can see here, D stands for dopamine, 5-HT stands for serotonin. The schizophrenia is primarily caused by imbalance of dopamine serotonin level in the brain. Our drug has potent activity for key receptors implicated in the schizophrenia, highlighted here. And then our drug directly modulate these key receptors or signaling cascades that contributing to schizophrenia. So dysfunctional this signaling cascade also known to cause imbalance in other downstream targets such as nicotinic, muscarinic, NMDA, GABA, as well as inflammatory cascades. Our drug has shown in various translational animal models as well, the activity for not only the main targets, but also dopamine serotonin targets involved in schizophrenia, but also downstream targets that significantly contribute to disease, such as inflammatory targets, nicotinic, NMDA, and then GABA as well. The combination of that one resulted in robust efficacy for major symptom domains of schizophrenia as well as neuroinflammation. I will walk through the key data. So now, before going to that, if you would like to assess how good is the drug for treating schizophrenia, and then also what's the top two unmet need? Of course, there are several unmet needs. If you would like to evaluate a drug, get a sense of comparative assessment, maybe the top two unmet needs to compare. The top two unmet needs are ability to treat negative symptom. This is one of the major symptom domain in schizophrenia. And then also the treatment adherence. As highlighted here, the treatment adherence is one of the major concern in the treatment of schizophrenia, because this is a lifelong treatment in most patients. If the patients are not taking medications, they are going to end up in hospital acute setup. So the treatment discontinuation attributed to the two features associated with treatment. One is suboptimal efficacy to treat major symptom domain. The second one is the poor tolerability and side effects. The combination of this could lead to treatment discontinuation. We call them as non-adherence. If you look at all the marketed drugs currently in the currently available as standard of care, around 30 to 45% discontinue medication in a short-term trial, and then in the long term, reported around 70%. So this is really a major concern. That means with the available treatments, we call them antipsychotics, more than 1/3 of the patients are not compliant to the medication. They are not taking medication, or they're discontinuing in a short term. So if you look at our data, our drug not only showed robust efficacy for negative symptom, that's one of the very hard symptom domains to treat, but also overall treatment adherence is almost 50% better than any other treatments currently in the market. That in the similar trial, while other antipsychotics showed 30 to 45% dropout, our drug has shown from the two large randomized trials, 12 to 16% dropout. That clearly an indication, to my knowledge, as a superior efficacy and then safety profile demonstrated by our drug. This is in a top level. One can assess this one, how good is the drug compared to other treatment options currently available. To support this, I will walk through some of the clinical data. So these are the clinical trials completed in the phase 2 to phase 3, and then last study we are in a process of initiating the study Recover 2. So these are required for NDA submission. The two large studies have already been completed successfully. The Refresh trial, global randomized trial, it was a dose-finding study with the 3 doses. 2 doses, low dose and high dose, showed statistically significant outcome. The middle dose also showed really robust outcome, but it fell short of statistically significant because it was a small arm study. That is understandable. However, if you look at in the second study, again, the top dose showed robust efficacy in 411 patients. Trial design was identical to the Refresh study. So the data generated in the Refresh study in 234 patients we were able to reproduce for the top dose in the Recover-1 study. So that gives us a significant confidence for moving this drug to NDA development. The long-term safety study is a one-time requirement. We are almost near completion of the study. As of today, we have close to 100 patients completed. In the next few weeks, we are able to complete 100 patients in this study. That is the requirement for NDA. So again, all 3 doses are generally very well tolerated, and then both in terms of efficacy and then safety. In Q4, we are going to complete this study. Now, the last study, registrational study, we are in a process of initiating. This is a required study for NDA, and then this is the last study. And then the study Recover-2 is identical to Recover-1, except the low dose is replaced by middle dose 30 milligram. Other than that, as highlighted at the bottom, the caption, we completed a long-term carcinogenicity study, drug manufacturing for NDA. All this process have been completed. So based on the development progress, we remain very optimistic and confident for driving this program to NDA in early 2026. So this is the phase 3 study design. It's a very standard design in the last 25 years, by and large for the primary endpoint, and then key secondary endpoints. The endpoints are kind of remain very constant. So the primary endpoint is total PANSS score reduction from baseline to end of treatment compared to placebo. And then key secondary endpoints are positive and negative symptoms. The remaining endpoints vary from trial to trial. However, in our study, all the secondary endpoints highlighted here, the study met both primary as well as all these key secondary endpoints. These data generated, the data generated in this study really makes this drug very robust and then, to my knowledge, superior compared to the standard of care. Now, here is the primary endpoint. This is an approval criteria to market for an antipsychotic. So we have seen in 4 weeks, 10-point separation from placebo. If you would like to, with the effect size of 0.6, this is really a robust outcome. If you would like to compare this data with other standard of care, how robust is this data? So to my knowledge, the widely used standard of care antipsychotics show in similar trial anywhere 3-point separation to 8-point separation. Having seen 10-point separation from 2 large trials in the phase 2 as well as phase 3, I believe this is really a robust and superior data. So this is about approval criteria to market. Now, having said that, the drug to be widely accepted by patients and their physicians, the key secondary endpoints are very critical. They are very critical for patients' recovery, relapse, and various other factors, in general, to have remission in these patients. So what are those criteria or secondary endpoints? So I'll quickly walk through. The positive symptom is one, every single antipsychotic showed really good data because the positive symptom is very close, outcome is very close to primary endpoint. However, other secondary endpoints, what I'm going to display, not all antipsychotics have, at least to my knowledge, showed statistically significant outcome. For example, agitation is one of the symptoms associated with schizophrenia. Not all antipsychotics have shown this data significant outcome. Our drug has shown statistically significant outcome, not only in this study, previously completed study as well. This is really a robust data. This shows the broad spectrum activity. The negative symptom is the most important endpoint after the primary endpoint, because to my knowledge, without negative symptom activity to date, no drug antipsychotics currently in the market achieved at least $500 million sales. So this is really a very critical endpoint. We use two scales to evaluate. One is Marder scale that is focused on 6-point outcome, very focused on negative symptom. Drug showed good negative symptom outcome. And then other one is 11-point outcome that to some extent comprising of cognitive function as well as a mood and then depression. So on both scales, it showed statistically significant outcome. So what we can take home message from this one, as I highlighted, this is really robust data. I will show you in a minute the biomarker data to further confirm negative symptom, good data generated here. So with negative symptom, robust activity, one can draw a couple of conclusions. As I mentioned at the beginning, negative symptom is the most difficult part to address. Without addressing negative symptom, patients are not going to fully recover. So having seen good negative symptom, this is an indication that patients are on a recovery path that already demonstrated by low discontinuation. And then also the negative symptom, good data, it's also an indication that it can limit or avoid relapse in this patient population. Symptom relapse is one of the major problem associated with these patients. And then this is a marker that patients, to a great extent, it can avoid relapse. Lastly, negative symptom activity is a key for developing drugs for beyond schizophrenia, such as bipolar and major depressive disorder. So this has a not only to treat schizophrenia, it's an indication that great potential to develop beyond schizophrenia to larger indications, such as bipolar, major depressive disorder. So social cognition and then functional outcome, PSP, personal and social outcome, these are other two additional secondary endpoints very closely related to negative symptom. When I say very closely related to negative symptom, without negative symptom, robust activity, one cannot expect to have a statistically significant outcome. This further supports negative symptom data. And then CGI is another one, functional outcome. Again, this is also to a great extent, not only the psychosis and functional outcome as well evaluates. As you can see, greater than 1 point reduction is equivalent to primary endpoint. And then we have seen 78% patients show statistically significant outcome, while other antipsychotics currently in the market in similar trials show 30 to 45% dropout. This further kind of attests the robust efficacy. So this is the summary of what I said: good efficacy for primary and then key secondary endpoints with good effect size. So now quickly switch over to the side effect profile and then surrogate endpoints. Why surrogate endpoints are important? So surrogate endpoints, it shows the comorbidity to address, as well as it further strengthens the primary outcome, especially the key symptom domain, negative symptom. Sexual function is one of the major side effects we see in this patient population, or morbidity. Around 60% women, 55% men suffer from sexual side effect. In this study, we have seen women patients show statistically significant outcome, improvement in sexual function. Men also showed improvement because in men, it takes longer time than women, because women are more sensitive to hormone prolactin. I'll show you in the next slide. So this data, statistically significant outcome, shows that our drug can address the significant unmet need, sexual dysfunction in this patient population, but also it further supports the strong activity for negative symptom. Without negative symptom, you can't expect to have good activity for sexual dysfunction or improvement in sexual function. So prolactin is another biomarker. It is very much related to decrease in disease, as well as a decrease in prolactin is related to decrease in metabolic side effects, as well as improvement in sexual function, overall improvement as well. This is really robust data seen on the day 14 itself, statistically significant outcome with this drug, while currently in the blockbuster drugs, some of the blockbuster drugs currently in the market are known to increase the prolactin. So here we have seen significant decrease. This is really a good outcome. So BDNF is a neuroinflammation related marker, or neuroplasticity, we call it. Again, this is the first time an antipsychotic showed statistically significant outcome. This is related to primary pharmacology. This can enhance the efficacy and decrease the side effects. That's what we have seen in the clinical trial. These two biomarkers are further supporting the robust anti-inflammatory effect because pro-inflammatory cytokines are highly expressed in this patient population. Dose-dependent decrease in pro-inflammatory cytokines, a first time ever reported in a total treatment population, statistically significant outcome to my knowledge. Now, this is the one very unique novel biomarker. Early this month, we had a KOL event. So this further supports the robustness of negative symptom activity with our drug. So what we have done here, so the biomarker speech latency is a signature mark in psychiatric patients, especially negative symptom. So the latency related to negative symptom used as a objective biomarker. So while every single schizophrenia patients have negative symptom as well as positive symptom, various other symptoms associated with that, how do we evaluate patients with a high degree of negative symptoms, whether drug is working in them or not? One way to evaluate, you take the total number of patients treated in the schizophrenia patient population, and then you use the digital or a vocal biomarker, and then see how many patients in that total patients enrolled have significant negative symptom. We call them at vocal biomarker positive patients. In the 411 patients, we have 220 patients have significant negative symptom component. That does not mean that remaining 187 patients did not have negative symptom. They also had negative symptom, but besides negative symptom, they also had various other symptom domains as well. So by seeing the activity in only 220 patients, which are significantly negative symptom, if our data shows robust activity, that further confirms the data what we generated for negative symptom. That's what this vocal biomarker data is about. Now, if you look at the data here, if our drug is working for negative symptom, has a robust activity, we expect to see in this patient population both the total outcome as well as a negative symptom data. As you can see in the left-hand side here, in a 220 patients, when we see the activity, as you can see, the PANSS score reduction in this came out to be within the group, almost like a 26, 27 drop, and the placebo adjusted came over 15. This is really robust data. And then if you look at the effect size, close to 1. While when you combine data, if you look at in the total PANSS score, it was a 10-point separation. That means our drug is working very well for negative symptom. And then another way to confirm this data further is you take out only the negative symptom subscale, where we have seen around 2-point separation from placebo, 0.4 effect size. If you look at here, only the negative symptom enriched patients decrease in negative symptom is almost 3-point separation with effect size of 0.6. So both in the total overall outcome as well as negative symptom, this further confirmed the biomarker data. This is really robust data outcome. Just to summarize here, the drug has shown robust efficacy for primary endpoint, key secondary endpoints, especially negative symptom, that is the top unmet need, but also the treatment adherence is much superior compared to any other antipsychotics currently in the market. And then efficacy further supported by both blood biomarker highlighted here, and then as well as a objective vocal biomarkers. So to my knowledge, the data presented here further supported by biomarkers. This makes brilaroxazine, a very robust drug. Now, in the interest of time, I'll quickly walk through side effects, very benign side effect, over 5% side effects, what we are seeing, around 6% headache, and then around 7.5% somnolence. But again, these two are very benign compared to other antipsychotics. Remaining side effects are, very benign. Now, if you compare the weight gain, overall in the study, there is no statistically significant outcome. But however, few patients in the US showed both in the placebo treatment arm showed slightly increase in weight highlighted here. But this weight gain is not related to mechanism to our knowledge. If it is related to mechanism, we should have seen increase in lipid profile as well. But lipid profile looks really good, decrease in cholesterol, LDL, and an increase in HDL. These are not attributing to weight slightly increasing weights in few patients related to drug, rather it is related to diet habit. Now, this is the drug-drug interaction based on the data generated here. Our drug unlikely to cause drug-drug interaction. And then while some of the blockbuster drugs, antipsychotics currently in the market in similar trial, reported to increase significantly drug concentration that sometimes in actual practice lead to decreasing dose level and that compromises the efficacy. And then causes side effect as well. Here, this is we expect to contribute significantly enhanced efficacy and decreased side effects. That's what we have seen in the study. Now, both good efficacy and safety profile lead to good compliance. 16% dropout versus 30 to 45% for marketed drug. And then there is no patient dropped out because of the side effect in the most efficacious dose, top dose 50 milligram. That makes this drug really robust efficacy and then good compliance. So this is the comparison data of phase 2 and then phase 3. We believe this is a very much reproducible in the trial because these two large trials show reproduced data. Now, this is the summary of what we have. Consistent wide spectrum efficacy from the two large randomized trial and trial conducted very well in the global level because it met every single secondary endpoints and primary endpoint, good consistency. And then also side effect and efficacy ratio looks really great. That's very important in the long-term treatment. So lastly, we believe this the our drug address a significant unmet need. As I mentioned at the beginning, top two unmet needs are ability to treat negative symptom as well as a major symptom domains, and then also treatment adherence addressed very well compared to other treatment options. This is the comparative data of brilaroxazine with other blockbuster treatments currently available, not only on the primary endpoints in the left-hand side, but also it is separated from key secondary endpoints as well. So with this, I pause here. And then again, as I mentioned at the beginning, we expect to complete the long-term safety study in the next few weeks and thereafter give the top-line data. And then we, based on our development timeline, in the next 3 to 18 months, multiple catalysts we expect. The earliest one is completion of long-term safety study. We remain optimistic of submitting NDA in early 2026. So with this, I pause here and then happy to answer any questions you may have. You can directly write to us on our website here, IR contact information provided, but also you can reach out to our IR colleague, Ben at Lytham. So thanks again. I really appreciate you all taking time to attend this presentation. Thank you, Ben.
All right, Dr. Bhatt, thank you very much for your time today. Certainly found it very informative. Before we wrap up, a quick reminder, anyone out there who's not already signed up for a one-on-one, please send me an email at shamsian@lythampartners.com. That's SHAMSIAN@lythampartners.com, or again, visit the conference website. Dr. Bhatt, once again, thank you, and we hope you have a great rest of the conference.
Thank you.